Welcome back to Chapter 3. This is the last part of Chapter 3 that we'll be talking about, and that is nucleic acids. So, nucleic acids are important because they are involved in genetics, or heredity. They store genetic information, they transmit it across generations, and they express hereditary information. When we say the word express, and we use that word a lot in this class, we say a gene is expressed. That means that it's involved in making proteins. Amino acid sequence of a polypeptide is absolutely determined by the inheritance of a specific region of DNA called a gene. Now, DNA is made up of lots of bits, but only the region called a gene is the region that will make a protein. So genes are made of DNA, and DNA is a nucleic acid, and we do have monomers of that nucleic acid called nucleotides. There are two main kinds of nucleic acids. There's deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA does provide directions for its own replication, right? So that's like how cells divide. We'll be talking about that at that point. We'll also be talking about how you pass information down to the next generation. That's why DNA is important, that it can make copies of itself. But DNA also directs the synthesis of RNA. We usually talk about it in terms of making messenger RNA or mRNA, though it makes all kinds of different kinds of RNA. And it's mRNA that makes proteins, and that whole process is called gene expression. Let me just take a quick look at that. Here we get to spend pretty much a whole chapter on gene expression. DNA makes mRNA, and mRNA leaves the nucleus in a eukaryotic cell specifically and goes out into the cytoplasm, or joins up with a ribosome and makes the polypeptide. Again, pretty much got a whole chapter on this, but for now I want you to remember that for gene expression, DNA makes RNA and RNA makes proteins. Let's take a look at the nucleotide. Remember, nucleic acids are polymers, and so we could say that they're polynucleotides, right? Because poly means many and nucleotides is the building block. So nucleic acids are polynucleotides. But here's the thing, and this is the part where it gets a little confusing, because the monomers are nucleotides, but nucleotides are complicated. There's three parts to a nucleotide. There's the nitrogenous base right here. And this is got a lot of nitrogen in it. That's why we call it a nitrogenous base. Notice that there are some carbons there that are hidden because that's what we do with carbons, right? We just don't show them in the cyclical molecules. So nitrogenous base. And then we have a pentose sugar. Pentose means 5. So 5 carbons, 1, 2, 3, 4, 5. I'm going the wrong way around the circle, but that's okay. Number 1 is right here, number 2 is right here. 3, 4, 5. And then we have the phosphate group right there. So nucleotides are complicated. There's 3 things to them. There's the nitrogenous base, the pentose sugar, and the phosphate group. Another thing to think about is this word nucleoside. And a nucleoside is the nucleotide that doesn't include the phosphate group. So this region right here would be called the nucleoside. All right, so if we look at the nitrogenous base really closely, we can see that the base has either 1 ring or 2 rings. And of course, as I mentioned before, they have nitrogen atoms in them. So these are the ones that have 2 rings. Those are guanine and adenine. And guanine and adenine have 2 rings, and purines are what we call them together as a group. That's a double ring structure purines. Pyrimidines, on the other hand, have only one ring to them. So cytosine, thymine, and uracil are all categorized as pyrimidines. We do shorten that to cytosine as C, thymine as T, and uracil as U. And purines are adenine A and guanine G. Also, interestingly, uracil, which we see right here, is found only in RNA, and thymine is found only in DNA, but the rest of them are found in both RNA and DNA. Now, remember I talked about there was a pentose sugar, and much earlier when we were talking about carbohydrates, I was talking about deoxyribose has an oxygen missing, and ribose is the basic pentose sugar. So if you look here, there it is. There's a ribose with the oxygen on the second carbon. And here's deoxyribose where that oxygen is missing. So it's going to be important to count carbons, and I've been doing that with you a couple of. So let's take a look at that again. There's the first carbon. Now notice right here we've got this little symbol, and it's one and then this little symbol, and that symbol is called prime. So the first carbon, instead of saying the first carbon, we just say 1 prime. And then this is the 2 prime carbon, and this is the 3 prime carbon, 4 prime carbon, and 5 prime carbon. So on this structure, the difference between ribose and deoxyribose is at the 2 prime carbon, right? At the 2 prime carbon. That's going to be a shorthand that we will use later in the semester, especially when we're talking about the way that the polynucleotide or the nucleic acid forms. So we'll get into this again in other chapters, but for now, let's take a look at the way that we have our polynucleotide here, and there's 5 prime and 3 prime. So what we're going to be doing is we're going to be having a connection from one nucleotide monomer to another nucleotide monomer between the 3 prime carbon and the 5 prime carbon. We'll take a closer look at that on the next slide. If we look at the nucleus side, there's our nitrogenous base connected to our pentose sugar, and that sugar can be either deoxyribose or ribose. And this nitrogenous base can be any one of the 5 nitrogenous bases that I mentioned on the previous slide. When you're connecting nucleotides, what you form is something called a phosphodiester linkage. This is the 4th kind of covalent bond that I've talked to you about in this chapter. Make sure you go back and take a look at those and understand the differences between them. So we're forming a phosphodiester bond. That just simply means that there's 2 ester bonds forming, and that means it's happening in the middle is a phosphate group, so it's phosphodiester. Notice that the 5th carbon, the one that's sticking up and that is attached to the phosphate group, is connected to the 3rd carbon of the sugar in the other nucleotide. So it's a 5-3 phosphodiester bond. The sequence of the bases, of the bases that are found in those nucleotides, it's the sequence of those bases that is the most important aspect of genetics. But of course, you can't have that genetics unless you have the linkages between the bases. The order of those bases that hang out, that's the genetic information. So DNA and RNA are very similar, right? They're both made of nucleotides. But DNA molecules have 2 polynucleotide chains that interact with each other. So we say it's a double bond or a double helix because they do actually spiral in space. This picture doesn't show you that. On the next page I have a picture of them spiraling, but a double helix. In the DNA double helix, the two backbones run in opposite directions. So in this one, we've got a 3' end down here, and on the other end of it is the 5' end. So there's a 3' end, and there's the 5' end. On the opposite one, on the one that it's connected to, the other polynucleotide, notice that the 3' end and the 5' end are on opposite sides from its partner, right? So this polynucleotide runs from 5 to 3, and this one runs from 3 to 5 as we go down the page. The word we use for that is anti-parallel. They are parallel to each other, but they run in opposite directions. If you're looking at a DNA molecule, they are very, very, very long, with thousands or millions of base pairs present. So this is where I'm showing you the spiral nature of the double helix, and the nitrogenous bases are paired up towards the center of that double helix. And notice they're connected by hydrogen bonds. So there's your hydrogen bonds right there, connecting the bases. And the trick to learn here is that adenine is always bonded with thymine, and guanine is always bonded with cytosine. It's called complementary base pairs. So we've got complementary base pairs, adenine, thymine, and guanine, cytosine. The blue ribbon that we were looking at before is known as the backbone, or you could call it the sugar phosphate backbone. The sugar phosphate backbone is really important because that's where those phosphodiester bonds are happening. That's what's keeping the polynucleotide together. But what's keeping one half of the double helix connected to the other half of the double helix is those hydrogen bonds. We can see hydrogen bonding in RNA too, and I just want to point out just very, very quickly in this kind of pale blue RNA molecule that if we were to follow this ribbon of blue all the way around, it would actually connect back on itself to back over here. So even though we have hydrogen bonding between base pairs in an RNA, the RNA only has one backbone. And that means that it is not a double helix, even though it looks like we've got some pairing going on here, which we do. It's not a double helix, it's just a single polynucleotide folded back on itself. So, and that's what I just told you on the previous slide, that RNA is a single strand, but that it can have base pairing. And remember that RNA doesn't have thymine. So the rule in DNA was that adenine bonds with thymine, right? That was the rule in DNA. But in RNA, the rule is that A pairs up with U. This is for RNA. And you can see that right over here. Back 20 some odd years ago, there was this huge project that took many, many years to complete called the Human Genome Project. And the Human Genome Project was the first time we ever looked at the sequence of every A, T, C, and G in a human being. It took over 10 years to sequence the single human beings. That was back in the early 2000s, in the late 90s. These days, you can sequence an entire human genome in just a couple days. And so that's led to a whole field of study of biological inquiry, and what we have is just a huge amount of data. Bioinformatics is what we call the field of studying biology, where we use computer software to analyze all that data, that just so much data out there. So we use bioinformatics. We use genomics to analyze sections of DNA that are called genes. Remember, genes are the things that lead to proteins being made. So it's a subset of the DNA. And then there's a whole field of research called proteomics, and proteomics is the study of proteins. So all of these things together make a really interesting field of study, including, and all of these things can be applied to many things, including paleontology, trying to figure out, how much DNA from Neanderthals are found in humans, especially humans of European descent. We can look at DNA in evolution, medicine, conservation biology, and also how different species interact with each other. So looking at DNA, understanding DNA is hugely important for understanding our modern society. Finally, we're going to end up talking about how DNA and proteins are important in measuring how fast evolution has happened. If we look at the linear sequence of nucleotides in our DNA and how that sequence is passed from parent to offspring, what we know is that siblings have more similarity in their DNA because they have the same parents and the parents have given them bits of their DNA. This leads to understanding that siblings have more DNA in common than cousins, for example, or completely unrelated human beings, things of the same species. We use that same logic to understand that two species that are closely related have more similar DNA than two distantly related species. So using this kind of molecular biology of bioinformatics and genomics and proteomics really does become extremely useful in understanding evolutionary kinship. We'll be talking a lot more about all of these molecules in further chapters. This is just your basic background. I hope that you got a lot out of this. There is a lot to learn from this chapter and a lot to familiarize yourself with a bunch of terms that you might not have heard of before. So I encourage you to spend some time studying for the test.